Hello, I am Giancarlo Vanini, and I am an assistant professor in the Department of Anesthesiology and a faculty member in the Neuroscience graduate program and the Center for Consciousness Science at the University of Michigan. It is my pleasure to interact with you by this pre-recorded presentation and tell you about our center and what is going on in my laboratory. The Center for Consciousness Science was founded by Dr. George Masur in 2014 with a mission of advancing multidisciplinary research, education, and clinical care as it relates to consciousness. I invite you to explore the center's website and get in contact with the center's investigators. The center is formed by a core group of faculty, primarily from the Department of Anesthesiology and the Neuroscience graduate program, with a common interest in consciousness. It also counts with several affiliate members from the University of Michigan and other institutions from all around the US and abroad, coming from diverse areas such as psychiatry, engineering, pharmacology, and music, to name a few. This integration gives the center and its members a unique advantage for approaching the study of consciousness by a diverse multidisciplinary group using a wide array of techniques ranging from molecules to human behavior. Again, explore and get in contact. As part of the educational and scientific mission, the center organizes an annual scientific symposium bringing together experts to speak about different topics such as chronic pain and consciousness, psychedelics, and in a future event, near-death experiences. I will now turn the page to tell you a little bit about what we do in my laboratory, where we use a systems neuroscience approach to try to understand how brain circuits regulate interacting states of sleep, wakefulness, anesthesia, and pain. To accomplish this goal, we use a combination of techniques, including behavioral assays, chemogenetics, calcium imaging, EEG analysis, AAV-assisted pathway tracing, and pharmacology. To learn more about it, please visit our lab's website. One of the primary lines of investigation in my laboratory relates to research to anesthetic mechanisms. We try to understand how general anesthetics reversibly suppress consciousness. This is a pictorial representation of the first public successful demonstration of general anesthesia in human surgery. It was conducted back in 1846 at MGH in Boston. And since then, and despite over 170 years of continued clinical use of general anesthetics, the precise mechanisms by which these drugs cause the loss of consciousness remain incompletely understood. One of the main hypotheses in the field was put forth by Ralph Leidig, who was one of my neuroscience mentors, and suggested that anesthetics cause the loss of consciousness by hijacking brain mechanisms that regulate states of sleep and wakefulness. This hypothesis has been supported by abundant evidence collected over the last 20 to 25 years, mainly based on CFOS expression, pharmacologic manipulation of different neurotransmitter systems in different brain areas, as well as non-selective lesions of brain areas that are involved in the regulation of sleep and wakefulness. Some of these areas are shown here in this schematic sagittal view of the mouse brain. I'm going to show a couple of figures from interesting studies, the first of which was published by Luo and colleagues, and in which they infused histamine into the basal forebrain of anesthetized rats. This pharmacologic manipulation caused this shift in the EEG pattern In this case, after saline, we see a typical burst suppression pattern, which reflects deep general anesthesia and, of course, unconsciousness. Histamine infusion, on the other hand, caused a more activated EEG state consisting in continuous slow wave activity. The second study was published by Max Kelt and his group, and showed that selective genetic ablation of hypocritinergic neurons in the posterior hypothalamus prolonged the recovery time from general anesthesia. Hypocretins, also known as orexins, are small peptides used by a small group, bilateral group of neurons in the posterior hypothalamus. These neurons and hypocretins are promote wakefulness, and these neurons degenerate, causing the clinical manifestations of the sleep disorder narcolepsy. This study is neat because they demonstrated that hypocretins are, and these neurons using hypocretins as neuromodulators are involved in the regulation of emergence from general anesthesia, which is substantially delayed in narcoleptic patients. Because of the relevance of the preoptic region in sleep generation, many of these studies focused on this area. One of these studies, and in my opinion, one of the most relevant mechanistic studies in the field, also came from Max Kelt's group. In this case, Jason Moore showed that GABAergic neurons in the VLPO, the ventrolateral preoptic nucleus of the hypothalamus, these neurons are presumably sleep promoting, and they are excited by the inhalational anesthetic isoflurane. I'll say that again. Isoflurane, a general anesthetic, activates sleep promoting neurons in the posterior hypothalamus. So together, all the evidence that I just reviewed strongly supports the shared circuit hypothesis. However, virtually all these studies are correlational in nature, and to date, there have, or at the time we started our project, there have been no causal investigations on the role of discrete brain sites that regulate sleep-wake states in anesthetic mechanisms. Another important structure is the median preoptic nucleus of the hypothalamus, and this is the nucleus that we explored in our studies. And the reasons why we chose to work and explore this nucleus is because one, it is critical for sleep generation. Second, it contributes to the regulation of sleep homeostasis. And third, by the time we started working on this project, there had been no studies looking at the role of the median preoptic nucleus in general anesthesia. We published this work early this year. This work was published in Current Biology and received a very nice editorial highlight. We tested the hypothesis that activation of sleep-promoting neurons in the median preoptic and ventrolateral preoptic nucleus of the hypothalamus would modulate sleep-wake states and alter anesthetic sensitivity. To start, we used chemogenetic stimulation of GABAergic and glutamatergic neurons in the median preoptic nucleus to test whether they altered sleep-wake states. To this end, we used a conditional expression of an excitatory designer receptor targeting GABAergic and glutamatergic neurons in the median preoptic nucleus. Here I'm showing EEG and EMG traces recorded during states of wakefulness, non-REM sleep, and REM sleep. And I hope you can appreciate that you can, by using these EEG, EMG signals, you can easily distinguish the three states. In addition, here I'm showing hypnograms after vehicle injection, the control solution, and CNO injection, which is the chemical ligand that activates the designer receptors. And these hypnograms show how the sleep-wake pattern is substantially altered after administration of CNO. What we found was that activation of GABAergic neurons in the median preoptic nucleus induced a significant increase in sleep, and with no change in EEG power. Based on these data, we predicted that activation of sleep-promoting neurons prior to anesthesia would increase the sensitivity to general anesthesia. To test the role of these neurons in general anesthesia, we injected mice with either vehicle solution or CNO 1 hour prior to the onset of anesthesia. Then mice were placed in induction chambers pre-filled with 1.5% isoflurane, which is a surgical concentration of isoflurane. Right after, we measured the time to the loss of writing response. We waited for 30 minutes. We discontinued the delivery of the anesthetic vapors, and we measured the time to the resumption of writing response. Both the loss and resumption of writing response are widely used, validated measures, surrogate measures for the loss and resumption of consciousness in rodents. What we found was that stimulation of GABAergic or glutamatergic neurons in the median preoptic nucleus had no effect on induction and recovery times with isoflurane anesthesia. We also tested lower concentration of isoflurane, 1.2%, which is a sedative dose of this anesthetic with no differences either. And then moved on and conducted a number of experiments to validate the designer receptor and viral vector in the preoptic area. First, we crossed our transgenic lines, VGAT-CRE and VGLUT-CRE mice, with another line that expressed green fluorescent protein. And as a result, we obtained 2 new lines expressing GFP in CRE-positive neurons, which in this case were GABAergic and glutamatergic neurons. By using these new lines of mice, we were able to verify that receptor expression, the expression of designer receptors, here indicated by in red in this figure, had a nearly perfect overlap with GABAergic and glutamatergic neurons in the median preoptic and ventrolateral preoptic nucleus. In addition, we used CFOS expression and verified that CNO administration induced a robust increase in CFOS expression in both GABAergic and glutamatergic neurons in the preoptic area, confirming that CNO administration caused, indeed, neuronal activation in the preoptic region. Next, we used chemogenetic stimulation of both GABAergic and glutamatergic neurons. I'm only showing glutamatergic neurons in this case. And we targeted the VLPO, the ventrolateral preoptic nucleus. This is a bilateral structure deep in the brain, near the base of the brain. And what we found was that chemogenetic stimulation of glutamatergic neurons increased wakefulness and decreased sleep with no change, again, in EEG power. The increase in wakefulness caused by this manipulation was quite a surprise because the preoptic area has been classically considered a somnogenic area. So in light of these findings, our prediction was that activation of these wakefulness promoting neurons would reduce the sensitivity to general anesthesia. Using the same experimental design that we used for the median preoptic experiments, we exposed mice to general anesthesia right after injection of CNO to activate these neurons. And we found that stimulation of neither GABAergic nor glutamatergic neurons in the VLPO altered general anesthesia. To conclude, we found that activation of GABAergic neurons in the median preoptic nucleus increased non-REM sleep. Activation of glutamatergic neurons in the ventrolateral preoptic nucleus increased wakefulness, and that activating these same cells had no effect on the induction and recovery time from general anesthesia. We conclude, based on this data, we conclude that preoptic neurons controlling sleep-wake states do not necessarily mediate general anesthesia, and that current correlative evidence for a mechanistic overlap of sleep and anesthesia might not have a strong causal significance. I don't want to leave you without acknowledging my funding sources and research team. This study was funded by a grant from NIH, from the NIGMS, and the Department of Anesthesiology. Here's a list of my lab here. My lab members, the names that you see listed in using bold fonts are those individuals that contributed to the study that I presented, including George Mashur, who is my collaborator and also a co-PI on my NIH grant. With that, I would like to thank the organizers for the opportunity to share my findings with you. Thank you for your attention, and I look forward to our Q&A session soon. Thank you.